So hello everyone and thank you for being here. It's really great to talk here about Laudaceae. I haven't talked about these plants here since 2010 more or less. Unfortunately, Hank is not here today and he agrees with everything that I will say. Everything. No. But we're still working in collaboration. Now I'm a postdoc here working with Sebastian and Christy in Weimania. But Laudaceae is always in the back of my head, so I don't think that I'm working in a different family. I'm just diversifying. So today I'm going to talk about Supraocotea. That is a lineage, a clade. That is comprised the informal group Ocotea, the Ocotea complex. That is comprised of about 950 species and 18 genera. So this clade contains the most species-rich genus in the neotropics, that is Ocotea. Here we have the distribution of Supraocotea in red versus the distribution of Laudaceae in this light blue. Supraocotea is mostly distributed in the neotropic with few species in temperate regions and around 30 to 40 species in West Africa and Madagascar. So this is a mostly neotropical group. So Laudaceae, as many of you know, has this bad reputation of being taxonomically complex. It's just a bad reputation. It's difficult to recognize genera and species. And if you talk with tropical botanists, they will start saying that it's because they are mostly large trees with small flowers. Here is a large flower in Laudaceae. The species, most of the species are bloom for only a few weeks a year. Sometimes take 2, 3, or even 4 years to bloom. And the flowers are displayed at the top of the tree. And for recognizing genera and species, we need the flowers. So that creates a kind of a difficult scenario. For instance, in this one, a large tree, mostly Supraocotea compound, so contains only canopy trees or subcanopy trees. Here we have a nice picture for a tree that I estimated 1.5 million flowers blooming at this time. And that tree bloomed only for 2 weeks in Panama. And when you are here underneath, you don't see those flowers. Sometimes you can smell it, but you don't see them. So that creates this kind of difficulty to collect them. They can be up to 40 meters tall, with small flowers on the top, so people sometimes miss them in the forest. But the flowers are kind of cute. So small flowers, tremendous flowers, and small flowers, that has to be always there. And they have this really interesting characteristic. With the stamens open these flaps up, and then the pollen gets stick to those flaps. Those are the flaps. Then we have these glands here at the base of the stamens and release this nice smell. I would say like a 90% of the time is nice, sometimes it's not that nice. But that attracts pollinators. So pollinators come and then they get stuck with the pollen that is already exposed in the flaps. Fruits are these small avocados. This is the avocado family. That's why it's one of the most amazing families. You don't like avocados, cinnamon. You don't like cinnamon, camphor. You don't like camphor, sassafras. You don't like sassafras. I don't know what is wrong with you. But in this clade, fruits are always has only one seed, and they have this couple at the bottoms subtending the fruit. So you get this combination of colors quite often, and that is to attract frugivores or to indicate signaling frugivores about where the fruits are. Remember, large trees in middle of the neotropical canopy, everything is green. So you signal frugivores with these things. So genera are delimited mainly in 3 floral characters. One is the number of stamens and can be 3, 6, 9, 9, 6, 3. Remember, this is a tremendous flower. the number of locules. Those are the flaps that move up. They can be 2, they can be 4. And sometimes the arrangement is also used to the limit of genera and breeding systems. If these are hermaphroditic or dioecic. Hermaphroditic having male and female organs in the same flower, and dioecic having females flowers in one tree and male flowers in a different tree. We have a gynodioecic. Gynodioecic means female flowers in one tree and hermaphroditic in another tree. I will explain this one better, and probably at the end of this talk you will be tired of listening about gynodioecic. So again, has this bad reputation, Laudaceae. And if you look at the late 70s, early 80s, what we had in Laudaceae was insufficient fertile collections. We don't have that many collections because people didn't collect them. We didn't have taxonomical keys, not for identifying genera, neither for identifying species. We have few monographs, few neotropical monographs. The first neotropical monograph in Supraocotea was published in 1982. And I think that we have like 2 more after that. We didn't have any way to recognize species at that time. So Hank here, when he was collecting in the 80s, probably he doesn't know that this picture exists. It's like kind of changing the reputation of Laudaceae. And the last 40 years, he has collected a lot of fertile material. He has produced a lot of keys for identifying genera and species. He doesn't produce that many monographs, but a lot of synopses. We will see why no monographs. He has described more than 200 species in Laudaceae and around 150 in Supraocotea. But still, it is really difficult to identify the species, but we are moving slowly to having a better taxonomy in Laudaceae. So based on this, on his work, and another hundreds of botanists who have been collecting Laudaceae regardless the difficulty that it has, we were able to produce a phylogeny, a molecular phylogeny a few years ago. This phylogeny is for 15% of the species of the clade. It's around 150, 57 species, and we include a species for all the genera. We use a different approach, a phylogenomic approach, using rat-seq. And we were able to finally resolve all these deep relationships. When I say finally resolved, because since 2001, when the first phylogeny was published here in Amsol with Andre Chandrabali, first phylogeny including species of Laudaceae, we haven't been able to resolve the deepest phylogeny using Sanger sequencing. So only until 2021, we were able to publish this phylogeny and resolve these deeper relationships. However, because life is not easy, so we found that no monophyly is prevalent in Laudaceae. It's not surprisingly, but it is surprisingly because most of the genera were not monophyletic, but some genera that we were expecting them to be monophyletic, they are not. So even we have more non-monophyletic genera than expected. Until today, we still have few non-morphological characters and not enough molecular information. Remember, this is only 15% of the clade, and nothing shows up like a monophyletic. No, nothing, a few groups, and I'm gonna talk later. So I think that this is a good point to say like my position about taxonomy. So my position about taxonomy is that it's a tool, communication tool, that should include good morphological characters, but also should talk about relationship. You have both. Having only one make it kind of like a loose wheel in the car. I heard an expression today. You said something with the wheels falling off the car, but I don't remember. But yes. So if you don't have all, there will be like a loose wheel falling out the car. So that's kind of my position. And then having all these limitations, we were in this kind of conundrum. What should we do? Should we start like naming clades? That's quite easy. You just name it. Clay with two species is monophyletic, or you put all nature there and it's monophyletic. Or should we wait until we have all the characters, good characters to resolve the phylogeny? Like how long it's going to take? 20, 40, 50 years? So what should we do? So we decide to do both, kind of do both. So we decide to have a multidisciplinary approach, and instead of just doing one of those, we decide to study ecology, evolution, and systematic in different ways in order to compile information that will help us in both ways. Having a better phylogeny and having a better morphological understanding of this group. So I'm going to talk about this approach that we have had for the last 10 years with Henk. And at the end of this talk, I hope that you can at least see some of the advances, the limitations, and the possibilities that we have in order to kind of understand the problems in the Ocotea complex and in Supraocotea in both. And also how we can use ecology and systematic and anatomy and many other tools in order to advance on these problems. So let's talk about ecology. So ecology of the Gynoraceae species Ocotea oblonga. So floral dimorphism is really common in Ocotea. Everybody knows that. Not everybody knows, but but now you know. So it's common in Ocotea. So species with dimorphic flowers are often described as dioecious, but a subset of species were described as dioecious having swollen pistil. This is in part as a consequence of implicit bias. So you have a female individual, you have to have a male individual, So they were forced to describe a species as staminate flower with a swollen pistil. They didn't consider other things. So in 1986, around 1986, Dr. James Lowen in Harvard University started seeing some collection with male flowers, so staminate flowers, that were producing fruits. That was kind of like uncommon. So, but he was using herbarium specimens. So he couldn't verify if this pistil, the ovules, were fertile or not. So he failed, or he couldn't say the species was dioecious or was Gynoraceae. Another species, and it's Ocotea tenera in Central America, Dr. Nathaniel Wheelwright, I think that I said the name right. He has studied this species, the ecology of this species. And sometimes he found a population with only, that looks like dioecious. And sometimes he found population that looks like hermaphroditic or Gynoraceae. So that means he decided to just call it dioecious. So he couldn't have a consensus if the species was really producing fruit for hermaphroditic individuals or for male individual with a weird mutation sometimes. So having this gap in the knowledge, we decided to move further and study this species, Ocotea oblonga, a species that was described from female individuals, and later on described as having male individuals with a swollen pistil. And we took advantage of this biological station, Barro Colorado Island in middle of the Panama Canal, when we have around 4 or 5 different plots, some of them dated since 1982. And when in 2016, we have a population of at least 300 individuals of Ocotea oblonga. So you have 300, at least you will see what is going on. So we decided to go there. We followed the tree for 4 different seasons, and we found that indeed female plants produce fruits, and hermaphroditic plants, now I call them hermaphroditic, so pollen producers, staminate flower with a swollen pistil, they were also producing fruit. So they were indeed hermaphroditic. So the species is Gynoraceae, not dioecious. However, because so we look at a morphological variation between the individuals, and what we found is that female flowers has a largest stigma in comparison with the hermaphroditic flowers. But hermaphroditic flowers are largest in average than the female flowers. And also we found that at least the population is hermaphroditic bias, the population in Barro, Colorado, not the species so far. That means that we have at least 4 individuals, for 4 hermaphroditic for 1 female. So you get more hermaphroditic in this population. But we want to look at the deeper ecology of this species. So we look, and we found that female individuals produce more fruits in average than hermaphroditic here. But when we look at the fruits, the mass of fruits, there are no significant differences between fruit or seeds between both genders or both sexes. So hermaphroditic and female, they invest the same in fruit. When I say invest, okay, one extra thing here. I'm trying to do germination experiments, but I was unlucky that during those years we have an infestation of these beetles that destroy hundreds of thousands of seeds. So I was able to germinate some seeds from both sexes, hermaphroditic and females, but not enough for doing a statistical analysis. So in the future maybe, but they produce fertile fruit or fertile seeds in both things. And when you look how they assign resources, so we look at historical data since 1982, and we couldn't find any significant differences in growth, in relative growth rate between hermaphroditic female and even the non-reproductive. So they're technically investing the same. Even when we include in the models, the number of fruit that they produce. So even if they produce a lot of fruits, they are growing in the same way. So what we can conclude from this one is that Ocotea oblonga, at least this species is gynodaisy, opening the door for exploring this character. We didn't see any trees switching genders or sexes during the time that we were there, and we didn't see a single tree containing both flowers. So they are females and they are hermaphroditic. The population is predominant hermaphroditic, even when the fruits are produced for females, and we didn't find any differences in any ecological differences between both genders, both sexes. I sometimes mess it up, but I think that Ocotea doesn't care about that. So let's shift a little bit gears here. So now we know that gynodaisy exists. We can see the ecology of this nice character doesn't affect, or there's no difference between different sexes. So we decide to say okay, that's another morphological character then. And then why we don't look the evolution of the characters that we already use in the taxonomy of this group, and how they can contribute to the taxonomical nightmare that everybody talks about. So using the same phylogeny, we decide to explore and do a ancestral state reconstruction for the 3 main taxonomical characters: number of stamens, number of locules, and breeding system, now including gynodaisy. So the reconstruction shows that 9 stamens in white is prevalent in Supraocotea, and we have multiple reductions, from 9 to 3 in purple or 9 to 6 in green. We didn't see a parsimonious progression from 9, 6 to 3. Something that is also interesting if you want to study gene expression or gene something. So you want to see how they turn on and off fertility in this group. But it's really, really interesting the way how they do it. And then what are the taxonomical implications of this finding? And then at least 2 examples. We can show that the genus Anaiva, we were kind of expecting it to be monophyletic. We have a species with 9 stamen in a different clade, unrelated clade than the species with 6 stamens. So Anaiva is not monophyletic. Okay, we can live without that. This is easy to take off, only 2, maybe 3 species. But then we have the genus Lycaria. Lycaria was this iconic genus in Lauraceae, like an easy to recognize, solid taxonomy we love, like Caria, and then species in 3 different clades. So 3 stamens evolve in multiple origins, multiple independent times. There's a monograph from 2000 in the Caria, and the genus was divided in 3 different subgenera. But when you look at the subgenera, they don't match the phylogeny. So even the subgroups that we can kind of organize using a regular taxonomy don't match the evolutionary history of the group. So there's a fundamental problem in how we define groups in Lauraceae. When we look at the other characters, locules, we only look at the number 2 versus 4. We didn't look at the arrangement. We found a similar story. Reduction from 4 in white to 2 in green in multiple origins. But also, we found some reversions from 2 to 4. So that's 2 instances at least in the 50% of this clade that we explored. And we have reduction from 4 to 2 and back to from 2 to 4. So yes, another plastic character that we used and we relied a lot on the taxonomy. And then let's go back to Gynolaceae. Now we know that exists in Lauraceae. Gynolaceae is often referred as a transitional state, an evolutionary transitional state between hermaphroditic and dioecious. So you get a hermaphroditic species and from some random mutation silence the male part of the flower, so it's female, and then nature takes it to be in dioecious. It's a really nice story. Sometimes it can happen, sometimes no. And then we decided, let's go and explore how this behaves in Lauraceae. I'm trying not to make too many jokes today. So we got there, and then we have another limitation. So remember that species are often described as dioecious in Lauraceae with a swollen pistil. Some species. So at this point, we have at least verified gynolaceae occurring in 3 different species: Ocotea tenera, Ocotea oblonga, and for personal observation of one of my collaborators in Colombia, in Ocotea infrafoveolata. So we have 3 species, and we are kind of sure that they were gynolaceae. But in literature, we don't know if the species that were described as dioecious, they are indeed dioecious or they are gynolaceae. So for that kind of limitation, we decide to have 2 different scenarios. One, using the 3 gynolaceae species that we know and scoring all the species, how they are scored in literature. And the other one is all the species with a swollen pistil described as having a swollen pistil, we call them gynolaceae. So 2 different scenarios. What is really interesting is that no matter how we score species, dioecious originated independently in 2 different clades. And if we use or we consider the species with a swollen pistil, gynolaceae, all of them fall into one clade. So we don't see the transition from hermaphroditic to gynolaceae to dioecious. At least we can say that we don't know if those dioecious species evolved from a gynolaceae ancestor, but the current dioecious species are not related to the current gynolaceae species. So maybe in the future we will have more clade with dioecious species, but for now we have independent origins. And again, no matter how we score the species. So something really interesting. Something else is that in this clade we have 3 informal groups: Ocotea smithiana, Ocotea minarum, Ocotea insularis. And another taxonomical consequence is that I think that was 2000, the last 2 years, Henk published this genus called Landea and converted one of those informal groups, the Ocotea smithiana group, now into a new genus with 25 gynolaceae species. So now we know that this is a system that is independent of dioecious, has a different evolutionary history, that we can use it as a taxonomical character now. And then last year, Dr. Pedro Moraes in Brazil described a new gynolaceae species. Then they use ITS, trying to see the relationship with other groups, and it fell into the same clade, into the same Gynoraceae clade. So now we have an extra line of evidence, and Gynoraceae tend to be related. We don't know if all of them, but most of the Gynoraceae species that we know so far are related, and then can be used as a new morphological character, so helping with the taxonomy. So what we can say is like a floral taxonomy character evolve multiple times, so everything is plastic, everything is like a Christmas tree, turning on and off in the phylogeny, and then we can contribute from the ecological study, doing an evolutionary study, with character like Gynoraceae to the limit monophyletic genera. So now I'm gonna shift to another area again. So I started looking at epidermal leaf cuticle when I was doing my master here. So we look at this amazing group called Ayuvia. I'm gonna talk about this later, but we didn't have a phylogeny at the time. So last year, kind of coming out of the pandemic, talking with Dr. Fabian Michelangeli, he proposed like, why you don't go back and look that in a phylogenetic framework? And then that's what I started doing, going back to the basics. So I looked for Ayuvia. Ayuvia is a really interesting group. It's the outgroup from my phylogeny, so there are not that many species here, but at least we know that species of Ayuvia are falls into three different genera, three different clades, so it's not monophyletic. One of the most important characters to differentiate Ayuvia is it has two locules. But in the last 5-6 years, something like that, there have been multiple taxonomical changes. So one of those was moving 3 species from Ayuvia into this genus Dambornegia, but also moving all the neotropical species from Cinnamomum and the monotypic Mosinoramne into Ayuvia. So we have species coming out and species coming in into Ayuvia. Still, we have some species of Ayuvia that are related to this other group, the Ocotea insularis group. That is one of the Gynoraceae groups. So still moving all these species around, we still have non-monophyletic genera. So with some other taxonomical implications. So you remember that we used to have this character to differentiate Ayuvia, so we lose it, because now each of these groups contains species with 2 and 4 locules. We know that it's not a good character, but still the taxonomy that we use today relied in that character. So now we are like in a kind of a genus that is not monophyletic, and now we cannot even separate many other groups based on that. But I don't have dogs on this fight, so I decided to look in a different way. So I started going back to anatomy, going back to doing epidermal leaf cuticles, but looking at in the phylogenetic framework, like I said before. So I think that I'm missing the slide, yeah. So what we found initially is that species from Cinnamomum and Ayuvia, they often have this stomata rim. The stomata rim is the cuticle when it gets swollen around the stomata. This is a really nice picture. It's a cutting stomata in half. That's really, really, really difficult to do. I have around 600 slides, but I have only one picture so far. So this is working in progress. So just kind of don't believe me, but believe me that I have seen these things. I will show you later. So we have this stomata rim, and we know what produces the stomata rim. Some species of Cinnamomum don't have the rim, but they have these papillae cuticles, and they belong to this clade, to the same clade. When we compare with the next clade, the clade in purple, we see that the stomata rim doesn't exist. Instead, we have this small structure surrounding the stomata. when you compare with the other group, the one that has the Burnegia and the species that were transferred, we have this other structure around. And that's what I said, and I have seen it, and I will show you later. This structure is not a cuticle. It's not the swollen cuticle. This is when the subsidiary cells push up the cuticle. So can look like, but they're two completely different formations. So in this case, we have three different clades where we can look at the anatomy and we can kind of get characters that allow us to plug species into the phylogeny without having the molecular. So I delete that slide, so that's good. So what happened here is like looking at this character instead of just looking for the phylogeny or looking at the morphological characters like the traditional ones. We were able to find a way, again, to plug species into the phylogeny. And why is that important? There are multiple initiatives now, at least we know about two, that want to create this large phylogeny in Laudaceae. Still, will be difficult to get DNA for all the specimens, and we don't have good morphological characters, to create groups into, at least into this clade that is Supraocotea. So using herbarium specimens, all these images are coming from herbarium specimens. We were able to find a different way to work around the classification that we have without changing names and without waiting for the big phylogeny to solve all the problems. So again, I don't like to be one side or the other. I prefer to start looking a different way, and that's what we did here. This is a work in progress, so hopefully by the end of this year or before some time, I will be able to talk more fluently and explain better, but this is a really good thing. We can look at different ways Laudaceae and still find a lot of characters. So, as a general conclusion, yes, epidermal cuticles can help, and we know those conclusions so far. Let's move on to this interesting part. This is the part that I was waiting to talk today. So, we have this problem with the phylogeny. We don't have enough characters. Again, I believe that we should have a taxonomy that integrate both, but what can we do while we are waiting for? And then this is the part that Hank support me, but he's not a big fan. So we decide to use the phylocode, or I decide to use the phylocode, and he say, okay, I cannot say no. So I decide to use the phylocode for naming clades in this group. So let me walk you a little bit. So remember, we have a phylogeny with 150 species out of 950 species. So this kind of 15% of the clade. With that 15% of the clade, we found that most of the genera are non-monophyletic, and then we were able to explore the morphological characters using taxonomy, and we know that they don't work. Just in this genus, Ocotea, Ocotea, has 450 species. That's around 45% of the clade. We use only 53 species. So that's 5% of the clade, and those 5% are in 7 different clades. Only 5% of the species are and we only know 15, and we already know that it's a mess, and we don't have morphological characters. But I don't want to be waiting for big phylogenies or for me to discover all the taxonomical characters, because I want to communicate with other fields, with ecology, with evolutionary biology, with people on the street. When you are flying, you can start talking with Laudaceae with everyone, so everybody hates you. So I decided using this phylocode, that is a topological system. That means we name clades independent of the linear system. So it's not affecting the linear system. And it's not hierarchical. That means that you don't have to name every single clade, and there's not a specific order to name clade. So it's a topological system. There are three different ways to name clades. One is node-based clade. So it's like when you're giving direction to someone. So you say the intersection of Kings Highway and Cho Boulevard. So that's what you are saying here. So you choose species to say like the most common recent ancestor from those two species. So you know the intersection. But sometimes you are not sure about that arrangement here. Maybe you are lacking species. So you have a pod sampling like us. So you can use a different way. You can say like along Kiss Highway from Cho Boulevard, South Cho Boulevard. So in that way, you are allowing the new species get plugged into the phylogeny, and you will still keep the same name. A different approach is when you have a morphological character, an apomorphia, and you say, for instance, Daisy. All the Daisy species will be named in this clade. So there are three different ways to name clades in this topological system. So we decide to at least have 7 different names, with 7 different, defining 7 different lineage. We choose 7 different lineage based on at least 2 conditions. They were well supported in concatenated and coalescent analysis for the phylogeny, but also that they were informative. They have something behind, not just a clade that we just want to name for fun. So one of the names is Supraocotea. That's where the name coming from. And now we know that include 950 species up to date, 18 different genera. So Ayuya is the Ayuya per se are the outgroup of this clade. But we also named this group Phylogeny. All these names were changes on this name were suggested by the co-authors of the paper. They didn't like the names that I put, but then we agree in simplifying the names to this one. So what's more complex than Phylogeny? So Phylogeny include all the species in Supraocotea that occurs in the Old World. So far, we know that all the species from Madagascar and the West Africa, they fall into the same clade, using ITS and using RAD-seq. So I think that clade deserves a name. And it's a clade that is inside Supraocotea. So we decided to name that. Another clade that is also based kind of a geographical distribution is Ocotea. This clade includes species from 2 different, this name includes species from 2 different clades, Damborneja and Electrifolia. And the distribution of those species mostly Mesoamerica, with few samples in Colombia, but no too far south on the Andes. So it's kind of, they get to the bottleneck in the Andes. So we decided to name this clade. There are some morphological characters for the whole clade, and some good characters that help to differentiate those groups. So probably this will be a different genus at some point in hopefully in my career. So the other 2 names that we decided to put was Diocotea and Pluriocotea. This is the name that they encouraged me to change because it used to be Pluriocotea. So they said, just shorten it. So it's Pluriocotea now. And then for these 2 clades, what defined was having as a criteria the breeding system. So all the Daisy species of this clade are here. And the Daisy species, or Gynodiaceae species, are in this clade. Something interesting here is now thinking about the linear taxonomy and the type species of Ocotea. What is the type species of Ocotea? And it's a Daisy species. It falls into this clade. So if I decide to make Ocotea monophyletic, that means that this clade has to keep the name. That clade contains around 100 species out of 450. That means that we need to give names, different names, to these 350 species. And we know that at least 50 species fall into 7 different clades. So where are those 350 species falling? No one knows yet. So that's kind of the limitation that we face now in Lauraceae. Now we know more or less what is going on, but we cannot just change name because pulling one name from one place will create a disaster in other places. So for now, the type of specimen is in the Ocotea. So, but why is the advantage of using phylocal? For instance, for me, the way how I've been using it, it's allow me to communicate, like I say, with general, with ecologists or evolutionary biologists, in the way that I can say Ocotea, they can go and collect species from Ocotea and get into that genus, but I can say the Ocotea. So they have the ability to go to the forest, find Ocotea, and they know that the Ocotea, so the dioecious species of Ocotea, is an independent clade of all the other Ocotea. So they are doing some linear analysis or any kind of analysis, and then they can plug relationships. They can start like having different groups. And if we are finding more groups and naming more groups, or even if you keep the taxonomy with no monophyletic genera, you will have the ability to point kind of sort of relationship among or within the genera. Doesn't have to be like that forever, but I think that for now allows us to move and communicate and just not saying that Lauraceae is difficult, so we don't give names to them, because that's a problem that we face every day in Lauraceae. So as a general conclusion, yeah, we find that Gynodioecium is a good character. In Lauraceae, of course, it turned into a taxonomically informative character. We know that the traditional characters has evolved many times, and we should think about something else. There are other characters, like using a leaf cuticle, that can help. And then, with the current information, we are really far away to have a morphological base and with a monophyletic-based taxonomy. And even what monophyletic means nowadays, I like to say that because it's kind of controversial, talking about integration and hybridization all the time nowadays. So who is monophyletic? We are monophyletic, and life is monophyletic. Everything else is boring. Then with that, before I have to thank a lot of people, especially because the talk is here. Technically, everyone who I look at here, I have to say thank you because it has contributed to the development of my career at some point. I want to thank all the botanists who has gone to the forest, collected samples, even when they don't know they are going to find a name, fighting with these big trees. But I need to thank Dr. Vanderwell, who allowed me to start working in Lauraceae when I was an engineer. I didn't have any idea what taxonomy was, kind of a little bit. Even Carolina Romero was the first person who explained it to me what a phylogeny was, how to read a phylogeny. So even from there, that started. Then also Liza Comida and Simon Quidbord, who were my advisors for the PhD, who doesn't work with Lauraceae, but they give me the chance to work with that. Michael Donoghue, also at Yale University, who allowed me to play in his lab. I felt that I was kind of adopted there. He just let me do stuff, and then how we create this beautiful phylogeny. Fabian Michelangelo in New York, who also took me back to the basics and say I need to work with leaf cuticle. I got distracted because I have to say thank you to Alba. So I will get in trouble. So Alba, thank you. And then I have to thank all the institutions who have contributed to this research. So this is the way how I start collecting nowadays. So because we are waiting for technology to solve the problem. So like having a drone, that will be nice. You fly a drone, but we don't have it. So I started using this big slingshot. Hank has been using slingshot, like small slingshot, but he has a person in Australia who he flies everywhere and collect for him. I didn't have the money, so I you know. So I kind of use my I don't know, some skills, like being lazy, a lot of duct tape, you know, and then I started using this big slingshot to collect samples. So I could reach up to 50 meters collecting this. This is a more sample, I can have better. Can do better, don't pay too much attention to the video in the weight, and it's kind of, I use a helmet most of the time. So sometimes I don't because I was kind of, for the video. But yes, I have to use a helmet, yeah. But it's not that dangerous. It's really interesting how. Of course, look for monkeys. I have problem with monkeys multiple times. They didn't like me. And then with this slingshot, I was able to collect sometimes just fruits or flowers, not always huge branches. You can break small branches, good enough for DNA and for the herbarium specimens. And again, I collected up to 50 meters in the canopy with this thing. So it was fun. Yeah. So, yeah. Yeah, so it's a long video, so people don't ask questions. Yeah. Thank you. Yeah.